This is an overview analysis of Dr. Cline's article presented by Amanda Ward and Colleen Valentine. The hypothalamus is located in the diencephalon and is one of the most important structures of the brain. It has a crucial role in appetite regulation. The areas for appetite regulation include the lateral hypothalamus, paraventricular nucleus, ventromedial hypothalamus, dorsomedial nucleus, and arcuate nucleus. The lateral hypothalamus has two neuronal populations that include melanin concentrating hormone and orexin expressing neurons. The ventromedial hypothalamus contains pericardia that synthesizes corticotropin releasing factor, which is a potent anorexigenic neuropeptide in chickens. The arcuate nucleus also has two neuronal populations, but with opposite effects on feeding behavior. It also contains a melanocortin system that is involved in regulation of energy intake and expenditure of melanocortins and AGRP, which is an antagonist of MC3 or 4 receptors. This proves to show the hypothalamus contains discrete populations of neurons involved in appetite regulation. Virginia lines of chickens were studied with low body weight and high body weight specifically, carrying anorexic and obese features over 7 generations. It was found that anorexic chickens typically don't respond to neuropeptide Y with increased food intake under a stressor, and high body weight chickens don't respond to recombinant human leptin with decreased food intake. The experiment measured the hypothalamic mRNA abundance of various appetite-related factors during a well-fed and fasting state in chicks. The low-weight chicks were utilized and they expressed higher quantities of mRNA encoding anorexigenic neuropeptides and associated receptors. In addition, the experiment measured mRNA expression profiles of appetite-related factors and individual nuclei within certain areas of the hypothalamus that were previously mentioned. The animal and experimental design consisted of closed-line populations of Virginia chicks, low-weight and high-weight chicks. The eggs were hatched in an incubator, and after 1 day, the chicks were moved to individual cages and fed a mash diet with tap water. After 5 days post-hatching, the chicks were divided into 4 groups that consisted of low-weight fed, low-weight fasted, high-weight fed, and high-weight fasted. The fasting occurred for 180 minutes. The chicks were then anesthetized and injected with RNA stabilizing buffer. After about 30 minutes, the coronal sections were collected in a cryostat and the samples of individual nuclei were collected. The next step of the experiment was to perform RNA isolation, reverse transcription, and PCR. The RNA was isolated from the nuclei samples via total RNA purification microkit and DNA Swan kit. The concentration and purity was measured by spectrophotometry. cDNA was analyzed using a high-capacity cDNA reverse transcription kit, and 5 of the isolated RNA samples were tested for verification. The next step was PCR, and the cDNA was used in the PCR analysis. Different primers were tested for genomic DNA amplification of certain genes represented in the nuclei, and some of the primers included CFOS, NPY, CRF, AGRP, CART, and more. A statistical model was created including the main effect of genetic lines, feeding state, and their interactions. It accounted for differences among nuclei so that they could be compared. In this experiment, not all targets were evaluated in all nuclei from the same sample. Factors selected were based on site-specific expression of hypothalamic neuropeptides and their receptors in different nuclei in mammals, as well as previously studied hypothalamic gene expression within the Virginia line chicks. The dorsomedial nucleus was analyzed for neuropeptide Y and MC3 receptor. There appeared to be greater mRNA expression in low-weight chicks than high-weight chicks in both areas. It was also observed that the mRNA abundance for MC3 receptor was increased when fasting in low-weight chicks, but not in high-weight chicks. The lateral hypothalamus was analyzed for neuropeptide 1 receptors 1 and 5, orexin, CFOS, and the mesotocin receptor. It was found that the neuropeptide 1 receptor expression was greater in the fasted state than in the fed state. The neuropeptide Y receptor 5 and orexin had the same effect, with an mRNA expression greater in low-weight chicks than high-weight chicks. The CFOS expression was greater in the fed state than the fasted state, and lastly, the mesotocin receptor had a higher expression in high-weight chicks than low-weight chicks in the fed state, but had similar results in the fasted state. The paraventricular nucleus covered areas including neuropeptide Y receptors 1 and 5, ghrelin, MC4 receptor, CART, and mesotocin. The neuropeptide Y receptor 5 and ghrelin observed greater expression in the fed state rather than the fasted state. The neuropeptide Y receptor 1, MC4 receptor, and CART had greater expression in high-weight chicks than low-weight chicks. And lastly, the neuropeptide Y receptor 5 and mesotocin had greater expression in low-weight chicks rather than high-weight chicks. Leptin was also observed to be negligibly expressed in nuclei collected, since there was no difference in CT nucleotide values between its cDNA and the negative control of reverse transcription in all of the hypothalamic nuclei. The ventromedial hypothalamus examined CFOS, orexin receptor 2, and LEPR. CFOS showed greater expression in its fasted state than the fed state. The orexin receptor 2 and LEPR presented greater expression in low-weight chicks than high-weight chicks. The RQ8 nucleus examined neuropeptide Y receptors 2 and 4, CRF receptors 2, AGRP, and MC3 receptor. The MPY receptor 2 had greater expression in the fed state than the fasted state, and increased in response to fasting in low-weight chicks, but not in high-weight chicks. The CRF receptor 2 showed no discernible difference in expression. However, fasting decreased expression in low-weight chicks, but it increased expression in high-weight chicks. Lastly, neuropeptide Y4, AGRP, and MC3R showed greater expression in low-weight chicks than high-weight chicks. The experiments that were run measured behavior parameters after the central injection of neuropeptides into 5-day-old chicks. Some low-weight chicks died shortly after absorption of the yolk sac, so severe anorexia was only able to be studied at such a young age. The results were difficult to understand because the amount and expression of patterns of appetite-associated factors in the brains of the genetic lines were unknown. In addition, the regulation of appetite is complex and has many factors and signaling pathways, which makes it hard to interpret the data. The gene expression screening that was run on the entire hypothalamus gives some clarity to the data obtained, but to obtain the best interpretation, knowledge at the level of the individual hypothalamic nucleus is needed. The screening of mRNAs encoding appetite-associated factors within discrete hypothalamic nuclei is one of the first conducted for any avian species. The results provided important information about the regulation and perikaryote expression. Also, the neuron has a unique anatomy in the site of neuropeptide Y synthesis, and the release is commonly far away. The fact that mRNA was detected in a nucleus does not infer that the transcript or the associated MPY is released in the same nucleus. There may also be other factors related to appetite that were not analyzed. One of the most understood systems involved in food intake is the neuropeptide Y perikaryote in the arcuate nucleus with projections to other appetite-associated hypothalamic nuclei. Neuropeptide Y was of interest because low-weight chicks was refractory to food intake after the MPY was centrally injected, along with exposure to a mild stressor during the early post-hatch period. Elimination of this mild stressor allowed the low-weight chicks to respond to exogenous NPY with an increase in food intake. There was a 3-fold greater expression of neuropeptide Y in the low-weight chicks than high-weight chicks in the dorsomedial nucleus, as well as upregulation in low-weight chicks, but not high-weight chicks in response to fasting in the arcuate nucleus. This data follows other reports that fasting increases neuropeptide Y expression, and that the arcuate nucleus is the primary site of neuropeptide Y synthesis in the hypothalamus. The increased expression of neuropeptide Y in fasted low-weight chicks could represent an attempt to compensate for the anorexigenic tone. The differences between the genetic lines were consistent with reports between genetically obese rats and mice and their lean counterparts, with a reduced hypothalamic expression of neuropeptide Y receptor 5 in obese Zucker rats and OB mice, and greater expression of neuropeptide Y receptor 5 in low-weight chicks than high-weight chicks in the whole hypothalamus. Based on other reports, it is possible that metabolic reserves in the high-weight chicks buffer them from the effects of short-term fast on energy balance. The results suggest that neuropeptide Y signaling in the low-weight chicks is intact but overridden by anorexigenic tone. The actual amount of food consumed by low-weight chicks is less than high-weight chicks, and this could be because the satiety-induced mechanism in low-weight chicks was more reactive, however more reduced in high-weight chicks. Lastly, the AGRP mRNA did not differ between the lines, which suggested that the nucleus-specific difference in the ARC is masked at the whole hypothalamic level. This experiment can be applied to humans with anorexia and obesity via the results of the low-weight and high-weight chicks experiment. This experiment can help us determine the neuropeptides and receptors associated with obesity and anorexia in humans, and therefore we can use blockage or controlled release of receptors and neuropeptides respectively in order to help treat the conditions of anorexia and obesity. This way we can also control the diet intake of patients. As for a critique of the study, more targets such as neuropeptides or receptors of the brain can be studied. This correlates with the next bullet point. In this experiment, only the popular gene receptors within each area of the hypothalamus were studied. And in further experiments, some unpopular genes could be studied to further broaden our knowledge of what pathways can be occurring. In addition, more research of the hypothalamus of chicks in general could help us understand these pathways and release of neuropeptides.